My name is Maxwell Wang, and today's topic will be transposable genetic elements, abbreviated as transposable elements. Transposable elements are mobile segments of our DNA that can travel from chromosome to chromosome and move freely within our genome. They were first discovered by geneticist Barbara McClintock during the late 1940s in maize plants at the Cold Spring Harbor Laboratory. McClintock's pioneering work transformed the way we think of genomes, because transposable elements are in the genomes of virtually all organisms on Earth, from prokaryotes to eukaryotes, from this mosquito to this boy to this bacteria. In fact, transposable elements make up 44% of our DNA. To put things into perspective, the DNA that actually codes for proteins within our genome only makes up 1 to 2% of our genome. That's crazy. That's mind-blowing. But before I begin, I'd like to mention that in this video, I will be assuming that you have an understanding of the structure of DNA, DNA replication, DNA transcription, DNA translation, and how gene expression is controlled. If you don't have a solid grasp on these ideas yet, or if you just need a quick refresher, please check out these wonderful Khan Academy videos before you continue watching this video. Alrighty then. So, scientists classify transposable elements by the way they move. There are two classes of transposable elements. Let's first talk about the second class of transposable elements, also called transposons. All transposons that are autonomous, or that can move on their own, encode for the protein transposase. Transposase is the protein that enables transposons to move. But not all transposons are autonomous. Some transposons are non-autonomous, meaning that they can't move. In fact, all transposons in the human genome cannot move. They either have mutations that hinder their ability to move, or are regulated and inhibited by some epigenetic defense mechanisms, including DNA methylation, miRNAs, etc. Here's the structure of a typical mobile transposon. The major feature you see is the transposase gene here. But surrounding the transposase gene, at either ends of the transposon, there are terminal inverted repeats. Terminal inverted repeats are sequences of DNA that are 9 to 40 base pairs long and that appear forward on one side of the transposon and backwards on the other. In this example, I've drawn the sequence here and here make up the terminal inverted repeats, as this sequence is just this sequence backwards. Terminal inverted repeats allow the transposase protein to recognize the transposon for excision. Transposons may also contain genes. If so, their structure would be like this, seen here. Now, since we know the structure of a transposon, let's see how it moves. Step 1. Transposase recognizes the transposon's terminal inverted repeats and proceeds to cut the transposon at both ends with blunt ends. These ends are called blunt ends in comparison to the staggered or sticky ends that have unpaired bases at the ends, as shown here. Step 2. The transposase finds a target sequence about 8 base pairs long in another stretch of the genome. Then the transposase cuts the target sequence so that each end has an 8 base pair overhang, as such. Step 3. Next, the transposon is inserted in the gap, and DNA polymerase and other enzymes come and fill in the base pairs missing. Step 4. Once the transposon has repeated this process again and moved on elsewhere, the target sequence will have a repeat. These direct repeats are the footprints of a transposon. Since the transposon is cut out and later inserted or pasted in another spot along the genome, we describe transposons as moving by a cut and paste mechanism, similar to the cut and paste function on your computer. So today we talked about the structure of Class 2 transposable elements and how they move. In the next video, I'll talk about Class 1 transposable elements, also called retrotransposons. In the third and final video of this series about transposable elements, I will talk about how transposable elements interact with other sequences of DNA and how transposable elements may have therapeutic value. I hope you enjoyed this video and learned a lot. See you next time.